So the question was, how do you calibrate the thermal camera to reflect the actual temperature? Because we are imaging the plants at such a close range, we don't have to calibrate the thermal camera at all. Basically, we just use its factory calibration. But I've seen applications where you need to calibrate the thermal camera when you put it on a UAV, because you're imaging the plants at far away, and the temperature can change after the atmosphere. So you need to put a special calibration board on the ground. But for our case, we don't need to calibrate the thermal camera.
So let's start off with a very general, big picture question, and that is that we have two very mature, well-developed facilities and a fledgling facility. and one of the questions is, what challenges do you have in these mature facilities, and what advice can you give, let's say, to the fledgling facility in terms of, what else do we need to do in these facilities in order to be able to bring about a real analysis of plant performance under different environmental conditions?
I think one important thing is, first of all, don't hurry up and give it a time, because I can only say, it took us quite some time before I even started with the phenotyping. I know that in Gattesleben, it took 3 years to get the first system really running in a routine, what you can call a routine operation. So it really takes time, first thing. Then secondly, probably you have done that, all the very basic validation experiments, like just take a whatever plastic plant, or take 8 plastic plants and put it to each of the chambers and let the robot run and see if you get the same results in all the chambers and something. This is what we all started with. And what we have done also is just have the very same genotype in all the compartments, see if it gives the same results. So getting basically from simple to a bit less simple and just validate it through. This is what we have been doing. And otherwise, just don't hurry up. And I think if you do that, really, you really do a very detailed validation, you will profit from that later on.
Maybe just to add a little bit about it is you really need to learn the limitations of your system, what it can and what it can't do. Because generally, when you read publications by other facilities, these are the success stories, because you just can't publish the, half a year of experiment that failed because whatever went wrong. So make sure you know where the limits are of the system, and also talk to other facilities that are already operating, maybe go and visit, and make sure that the staff you hire potentially in or that's working on it they don't only have the technical skills but also the soft skills to work in such a collaborative team.
So I guess part of what I'll say is that some of what it is, especially that Bettina just said, is the reason that we invited both of them to come here and see Envira Tron is because we suspect that someone that's been working in this area previously is going to be able to give us all kinds of pointers. So we have expectations for you this afternoon.
Plant breeders are forever telling us, that the data that we get in growth chambers doesn't really reflect what's happening out in the field. So what can we do with respect to these controlled growth conditions to provide information that would be of greater value to plant breeders and may better reflect what is happening out in the field? So I was interested in what Astrid was pointing out with respect to showing multiple plants together, growing in a growth facility where you could maybe test the competition between plants. Is that a possibility?
I think people are doing this in similar systems. At least I've seen, but this is, I guess, more like an ecological type of study where people are doing this in these large growth containers to really look at like competition of different plants in one container, for example. That's not what we would actually like to do. So we want to really focus, on crop performance under fluctuating conditions in our new growth facility, and we think that this makes perfect sense. Also, I mean, I'm not a breeder, but at least I think it would give some valuable insights into what breeder can use in their breeding pipeline from several perspectives. I mean, first of all, we can control fluctuations in a way that we can really understand and analyze the dynamics on a molecular level, which you would not be able to do in the field. So in terms of a basic research approach, this is very valuable. And if you really find new candidates, new processes which are underlying, for example, acclimation to fluctuating light, whatever, then this could be introduced into a breeding pipeline later on. So I think, from my point of view, it makes perfect sense.
I guess the other advice would be to become friends with a crop modeler, and make sure you work closely with them. So what we are trying to do in the controlled environment is not necessarily replicating exactly the field. I mean, we can certainly do that with simulating different climates and so on, but they will it's never going to be the same. The other thing to always keep in mind, the field is not the field. I can do an experiment in 1 year and do the same, location the following year and get completely different results. So what we're trying to do generally is work on individual traits that you wouldn't be able to do in the field because they would be drowned out by noise and just a too big G by E interaction. So work on component traits and then validate them in the field. And this is where crop modeling can then also help you, because we've identified, others in Adelaide have identified drought-related QTL, and they are beneficial in one environment but detrimental in the other one. And this is where crop modeling can inform you about, is this beneficial in a terminal drought scenario, or, if there is heat stress early on, and then almost help you hypothesize about some different ideotypes that might be different for different climatic scenarios.
So I don't have input on that per se, but I have got sort of an idea that plays into this question. It seems like to me when we have these different controlled environment phenotyping facilities, and they each have completely different benefits and things that you can do in them. So for example, at the hall that's at IPK, they can use lots of plants, whereas at Envira Tron, we can't do lots of plants at one time. We can do many environments, they can do fewer. So I imagine that there's the possibility that together we could do some coordinated experiments where if we were collecting data in all of these different ways, that we could make use of the benefits of all of our different facilities. I'm wondering what you two think of that idea especially.
Yeah, coordinated experiments are always fairly challenging. Yeah, no. They're within the EPPN, so the European Plant Phenotyping Network. There was an attempt to have the same maize lines grown across all the different facilities and have it as standardized in terms of how we grow them, how we measure all the data. It's not a trivial exercise. In fact, it's a massive challenge. And even having said that, there is a study published for Arabidopsis where they shared the potting mix they used, where they used the same type of growth chambers, supposedly the same climate conditions in all the different growth chambers, and got completely different results. So yes, you can definitely try and cross validate and cross-compare. But you have to realize that it's not necessarily gonna make you a terribly happy person, because sometimes it can be very challenging. And Astrid was part of that experiment as well.
She's smiling.
Do you want to comment on why they got different results? I mean, I think it's going to be very
Look, I can only speculate, but even if you have the same growth chamber from the same company, depending on how long the lights have been in there, there might be slightly different light quality. When the sensors were calibrated last, the humidity and temperature may not be exactly what you think it is. There's always operator influence. Yes, they were all supposedly watered, in the same way, but even water temperature can have an influence. So there are so many different aspects that can influence your phenotype in the end. So it's probably best to read the paper and see what. I mean, there were some that were comparable, but there were also individual locations that just stood out in terms of not fitting in. And for example, when we did the EPPN, they plotted the VPD at the different locations, and you have Europe, and then you have Australia somewhere down in the far left bottom corner. Because yes, it was a rather dry summer, obviously, and even in an air conditioning greenhouse, your humidity is nowhere near as high as it would be in Europe.
This makes me think of early days of transcriptomics.
So I think we should really not repeat the experience that we have made with an EPPN, but I think it's never possible to. I mean, and this was also not the intention in EPPN to have everything similar or everything the same way. I mean, it will never be that people would use the very same soil, for example. It just doesn't work out anyway. So I think it would maybe be a valuable cross-platform comparison. To look at one scenario, like for example heat, and we could perform it at one time point, at one key time point at development, and you can compare many different. You have many different chambers where you can do. Could look at different time points. This would be something to maybe think about. I mean, terminal heat, where that has an effect, but then deviate from this terminal heat to whatever other interesting or critical developmental time points and compare the effect of that, for example. This would be something where we could compare, like our facility, which would have we could grow plants longer, we could go into yield determination, for example, but you could compare many different time points because you have the different smaller facilities. So I don't know. This makes sense.
So we have a question online here. The question is, can you talk a little bit more about your pipeline for imaging, image data analysis?
So we at IPK, we have a self-developed pipeline for some very important reasons. So we wanted to be independent from the company because we want to be able to extract new traits for specific questions, which would be very time-consuming and expensive if you would ask the company. That's why we have our own pipeline. And on the other hand, we also wanted to be able to phenotype, or we are phenotyping many different species, under many different stresses or conditions. So the spectrum of architectures, plants, colors, for example, is very diverse, and so we need to have a very general image analysis pipeline that we can adapt to this variety. But But otherwise, I think it's a very general procedure that we all go through segmentation, which is one of the key steps in image analysis. We all have similar things developed to increase the quality of segmentation. And then for feature extraction, we all go towards machine learning or deep learning, I think. Especially for root phenotyping, this seems to be very important, and also hyperspectral imaging. Not sure if this was did cover all the complexity. I give the mic to Bettina.
So, Bettina, we want to hear from you, but also I would like to hear from Marna at the end of this question on what it was like with Envira Tron, because I know that we have almost nothing, and so I know it was hard.
So we, not like IPK, currently don't have sort of a fully automated pipeline. There's still a fair bit of customization for each experiment. Because we operate as a service facility, we may have from one day to the other come someone who's like, with a completely new crop species. So we try and be as flexible as we can there. We use a combination of sort of proprietary or, licensed software, own scripts. We're also now trying to benchmark against some of the plant CV algorithms available. And then for the more complex area of 3D modeling, we work with computer vision, and they use C++ and other coding software to really code and develop pipelines for quite complex analysis. So it very much depends on what the traits are you want to measure, what the species is you're working with. Some are a lot easier than others. Chickpeas, for example, have pretty small, fine fingered leaves. Or a rice plant in its early stages is something completely different to a maize plant at 6-leaf stage. So at the moment we don't have a one-fits-all pipeline, but really do it on a case-by-case basis.
So as Carolyn said, our imaging pipeline is in development, and so for me it was a very manual process. So being very early in the beta testing of the facility, I think I've become very aware that this was a great learning experience, but any data that we pull out of our experiment really just feeds into us doing it again, I think, based on what we've now learned. And so in doing the data collection from the thermal images, right now, when I showed you those images of, drawing lines, I think machine learning hopefully is the goal where this type of manual analysis will be much less necessary. But there was a lot of drawing and clicking and eventually, hopefully, training other people to draw and click, or wait until we have those tools. But Scott has been fantastic with building some tools to look at the data just from what we can do right now, and so that has been very helpful. I was excited to be able to look at the data and start to think about it, which I hadn't been able to do earlier when we conducted the experiment first. Is there more you want to know? Okay.
Fine. So early on, what I heard was curb your enthusiasm, slow down. And I'm delighted to hear from especially both Bettina and Astrid that this thing that we worry about, think about with this type of analysis, that even after you've been thinking about this for a longer period of time than us, it's not like it's a solved problem, which that's probably the wrong way to say it. I'm delighted that it's not a solved problem. I would love it if it were a solved problem. But it sounds like we're all in a similar boat.
Continuous process.
Yes, nicely put.
One thing that would be very useful to us, with respect to imaging analysis and that sort of thing, if there could be some sort of sharing of training sets, establishing, some of the training sets is represents a considerable amount of effort just to establish those. I'm not sure whether they would be shareable or not, but particularly for us in analyzing, maze structure, looking at the top down rather than looking at side views. If we had, some training sets to be able to do some machine learning with respect to those kinds of images, it would be very useful to be able to share those and make that sort of thing work. Go ahead.
So just with respect to sharing of data sets, so I think this is, I have to answer with talking about myopy and fair data. So I mean, we have published some data sets already, which it's Arabidopsis so far, but I know, I mean, we will publish soon data sets also for maze, for example, in our in-house database, but they will be open for everybody, and this will always include raw images and result images, and also the data data extracted with our software. So this probably would be useful then for you to look at that and to benchmark your software against these data.
There is already a maze data set publicly available from University of Nebraska that you can download and work with. And then also within the IPPN, there is the imaging working group who sort of co-organizes a conference each year where they put out a data set that then the different teams can sort of analyze to see who can come up with the best algorithm. So there are some data sets out available, and I think most facilities, even if they don't necessarily have them publicly available yet because they may still be in the process of publishing, I think if you approach them, most would at least give you the sort of anonymized data sets, in the sense that you wouldn't have all the full metadata associated with it, but at least the images to work with.
Yeah, this is kind of a general question, but so far all the high throughput phenotyping methods are using sensors. A lot of them are taking images, others are measuring external characteristics of the plant for the most part. But there's a lot of interesting and important processes happening within the plant that we also need to measure, whether it's biochemical or metabolite. Are any of you aware of high throughput methods being developed to measure internal phenotypes of plants?
Okay, shall I answer? At least, so I know that, I mean, people are now more or less routinely using hyperspectral imaging, and at least for elemental composition, for example, you can use, so it has been published that you can use hyperspectral, certain wavelength ranges are predictive for certain elemental compositions or concentrations. So I think it's much harder if you go for metabolomics and transcriptomics. I think this is not, I mean, you cannot derive this from any kind of imaging. So you will have to sample and you will have to do integrated analysis. I think what is really nice, what you have already or will have, automated sampling. I mean, this is something that from my point of view, will be very important in the future, because this limits for us, for example, the throughput a lot, because you will always have to go there to transport the plants to the point where you take the samples. Then you have to be really fast in order to not exceed this time window of whatever 1 to 2 hours in which you should sample all your plants. So if we could automate this sampling, then this would be much easier already.
Yeah, I mean along the same lines in terms of the hyperspectral imaging. So we have our system fairly recently, and what we are now trying to do is can we get good correlation with some of the biochemical traits we are interested in. So one being water-soluble stem carbohydrates, for example, which are important in drought adaptation. Most likely, we will not necessarily be able to measure the compounds themselves, but rather what a plant with a certain profile looks like. And that is using machine learning approaches to try and correlate the wet lab analysis with the hyperspectral images that we get. So that, in terms of high throughput, that is probably the one that has most potential, but it has its limitations. It's not a magic box that will tell you everything about the plant, and unless there is a signal coming off that you can detect, you would struggle to use that.
So David, as you know, there also are a number of sensors that are ones that penetrate the plant that are being developed by different people, and certainly it seems like at all of these facilities, those kinds of sensors are something that if we put those on the same plants that we're otherwise measuring, it'll be nice to be able to get, for example, what the nitrogen levels are of the plant, or what sugar levels are in the phloem. So those kinds of things, I think, as we go along, are likely to get more and more coupled with what we're doing with the kinds of things you saw today.
Carolyn's talking about the work of Leon Dong here at Iowa State, who's developed this nitrate sensor that was able to assess the nitrate level that's going through the vascular system in plants. I've tried to encourage him along the way to develop other sensors, that would, be able to sense various organic compounds by having an immobilized enzyme or something that binds, a particular substrate, et cetera, and gives you an electrical signal. Those would, I think, be very exciting to do, to give you sort of a real-time metabolic profile of, what's happening with respect to, certain other compounds. Any other questions out there?
Yeah, so I was curious about how exactly the IPK facility does their Rhizotron and their root phenotyping. Also, the climate control system in the larger pots was really interesting too.
Good question.
Thank you very much for this question. So we will have a kind of complementary root phenotyping infrastructure, which will cover the different systems that I've shown. So in these LimnaTech type plant 2 sensor systems, for example, we have, so I showed, no, I cannot show. We use in the small system, we use transparent pots, magenta boxes. So we just grow the plants at the side of the pot, and we see the roots growing along this transparent surface. We do imaging in the longer UV wavelength range, because this turned out to be very effective for that. It has a disadvantage because the pot surface is still straight, so it's not inclined, so you will only see a portion of the root there. But this is combined with shoot phenotyping in the automated system. In the large plant system, we are using, like it's in the very beginning, we are using larger pots, self-constructed pots actually, custom-made pots, where we have already an inclined surface. I mean, you can. I don't have the pictures of the systems here, but so we have. You can see, like in the Arabidopsis up there, it's very hard to see a root. I mean, there's a part of the root here, down here. For maize, so in the large system, it looks much better, because here we, as I said, we use a custom-made pot which has one inclined surface, and this inclined surface is made of a filter material which would not leave through visible light, because roots want to be in darkness. And in the small system, we use magenta boxes, but they are shaded from the light, just taken out manually, basically, for the imaging. But in the large system, we have this filter plate inclined, made of a filter that is, as I said, not transmissible for for visible light, but for near-infrared light. So we have an imaging system which has near-infrared LEDs and a specific camera sensor that takes images through this filter plate. And this looks basically very nice. I mean, I can show you something on my computer later if you want. So this is the systems in these conveyor belt systems. Then we have in the plant cultivation hall in the new building the Rhizotron system, which is again working with inclined surface, plexiglass surface, so transparent, but a bigger area to look at because it's about 90 centimeters in height, 70 wide. So it's quite some area that you can image the root. And in these containers, we will only have like transparent inlays that go at various depth into the soil, where you can insert small imaging and illumination units to at least have information about rooting depth. Yeah, so these different systems are also very different in terms of the number of traits and the precision that you can quantify the root in. Yeah, so these tubes will only be rooting depth. Rhizotron will be much more detailed in terms of root system architecture.
Any other questions out there? Yeah, there's a question back there.
I had a question from Bettina's slides. So you were using the probabilistic models to figure out what the most probable orientation of the leaves were from the 3D data. So is information shared between time points? So can you tell which leaf at one time point is the same as a leaf in the next one?
So the answer is yes. So you use the time course information and you know how many leaves were there the day before. So you obviously know that you use that prior information both about the number of leaves but also the leaf length. So obviously new leaves appearing is always something you then have to, incorporate into your model, but you also know that leaves generally get longer rather than shorter, obviously, unless, you have some mishap and they get ripped off. So if you know that leaf 1, 2, and 3, for example, over the last 2-3 days haven't increased in size, then you can reasonably easily assign them on the following day, and then any leaf that are continuing to increase in size are the new ones. So yes, you use the information from previous days.
So once a day seems to be often enough that you can do that with pretty high accuracy?
Yeah, I think with that initial study, it may have even been only every second day, to be honest. Yeah. But yeah, once a day is currently sufficient, yeah.
Well, registering images actually is a big challenge, and it's a big challenge even if you're taking, pictures with different modalities at very similar time points. So we're not even talking in a time course or anything of that sort, just simply trying to put together an RGB image with a thermal image and making sure that you're appropriately registering is turning out to be if you're trying to do that down at the pixel level, it's really problematic. So I have another very major question, and that is that Carolyn publicized this as a Women in Science activity, which I think was great. Talking to my colleagues in engineering, they're always complaining about the fact that, there are very few women who go into engineering careers and even to computational careers. However, in the plant business, particularly, people who've been funded by the National Science Foundation in the U.S., there's a lot more women now than there are men. So I'm wondering if this is a way of being able to attract more women into engineering and to computational areas by having this link, if you will, between plants and some of these engineering systems, such as the controlled environment systems. Any comments?
My first comment is that I see two young ladies sitting in the audience who are working in this general area for predictive plant phenomics. And I would ask you two, do you think events like this are worthwhile in that respect?
So having things like this means a lot to me, and it makes me want to do more things like this, and hopefully makes other people want to do it too.
Yeah, similarly, that was part of why I wanted to come. And I sad she left, but another woman in my lab who's actually a member of material science and engineering and working on these similar projects was here earlier. So, but she had to leave. But yeah, I mean, that was part of why I came to this, is I saw the woman in science. I'm like, oh, it's actually women speakers for once? Oh, that's exciting. So it's really satisfying to actually see that.
So there are a lot of data out there that show that when you see somebody like you working in an area, that you're more likely to feel welcome in that area. And I can honestly say that personally, I've looked back on why it is that I do what I do. And I think that it's because I am so completely clueless that I wasn't even thinking in that direction when I was younger. It didn't occur to me. And that apparently is uncommon for it not to have occurred to someone, that when they look out, the people don't look like them.
Okay. So I think I also got into this field rather accidentally, so I also started out with basically studying something similar to biology, as probably at that time many women did, because it was nutrition sciences, and it was like 99 women and 2 men. And but then I went into plant science, at some point I went into bioinformatics, and was with a lot of men, but I was at that time the communicator between the biologists and the bioinformaticians, which was something that was very productive, and something that also is probably part of my success now, that I can somehow, I don't understand every bioinformatician in very much detail, and I also don't know every detail that the engineers know about the facilities that we are using, but I probably, or I'm convinced that I know what is needed to understand how it works, and how to design these kind of systems, and how to make them, how to apply them, use them successfully.
Yeah, so I guess for me also, my degree in biotechnology, there was a reasonable amount of engineering, in that case process engineering, involved. We also had some basic level of computer programming and so on. And we were actually, our specific year was outnumbered for the females, but generally was a fairly balanced study. But now I guess, yeah, my role as well, similar to Astrid's, it's often as the interpreter between the different disciplines and domains, and making sure that the engineers understand what the plant scientist is trying to say, and that our horticultural technicians are aware of what's important for our mechatronics engineer to maintain the system, and vice versa. So I think it's bringing that basic interest and knowledge in engineering, and yeah, using that to bring the different disciplines together. Yeah.
Maybe another general comment, at least speaking for Germany, it's a general problem which is maybe more visible in engineering, so sciences, but it's a general problem to combine work and life as a woman with kids, and this is at least when you're in a leading position, it's always difficult, independent of the field that you're working in, and this is something that has to be improved through whatever political initiatives, and also changing minds, many minds, I think.
Marna, do you have?
Thank you. So I think it's interesting being in female in science. I guess my perspective is kind of a fun one because I am in a building that does biorenewables research, and I'm on a floor where I am the only faculty member from the genetics department, and the other faculty members are all from chemical and biological engineering, which has been a lot of fun. And the really neat thing is that on that floor, unlike most engineering floors on campus, we have 3 female faculty members, and for a while the whole floor was female faculty, and so that was pretty abnormal. We now have a male faculty member as well, but so I think that was kind of uncommon, but pretty cool.
So beyond my earlier comment about saying that I was sort of clueless, and I've always been sort of clueless, I still am. I will tell you one quick vignette, and that is that I can remember when I was a postdoc going into a meeting that was a bunch of people that were working on plant genome databases. And I sat down next to a friend, and there were probably 20 people in the room, and it occurred to me that I was the only woman in the room. And I can remember turning to my friend and saying, "Whoa, I'm the only girl in the room." And he said, "There's more than there usually is."
Hi, I'm Trina Dobbs. I just wanted to add to this conversation with a shout out to the NSF. I started out in engineering in 1977 at Georgia Tech in electrical engineering, where the ratio of males to females was over 100 to 1. I ended up not staying at Georgia Tech. My identical twin sister did, and she worked in EE for many years. I turned to biology, but over those many years, so little has changed. And the little that has changed with regards to women entering faculty and leadership positions, I give a lot of credit to the NSF. I think a lot of the reason plant biology has so many active female scientists is because the NSF mandated that everyone had to have a fair shot. And you don't see that at the NIH, frankly. And I don't know about other funding institutions, but I think it was a concerted effort by the NSF to make sure that women had the opportunity to compete fairly for grants and to overcome some of the difficulties that were mentioned with work-life balance, because women enter science in high school and undergraduate school and graduate school and even as postdocs in the biological sciences in equal numbers to men. They fall out when they get a tenure track faculty position. So anyway, I would just like to thank the NSF, and I think the NSF is largely responsible for what's happened so far, and it has demonstrated that being aware and taking steps to make sure that their equal treatment of different genders or underrepresented minorities makes a huge difference.
I think that's a great point, Trina, and a neat project that I'm on is a collaboration with 2 faculty members from the University of Minnesota. And so there are 3 PIs on the project. We're all females. And we are also, our broader impacts are all about engaging females in agriculture careers. And so the NSF has been very supportive of those efforts, and I think that's a really neat aspect of the project.
Well, let's thank Carolyn and her crew for putting on this symposium. So it's been a great experience.